I am honored to be provided the opportunity to present this research at the 2020 CUNY Research Scholars Program Virtual Symposium. I, Gearoff Cruz Rodriguez, a member of the CRSP program at BMCC, have done this research under the mentorship of Siddharth Ramakrishnan, Director of Research at BMCC. In this presentation, we'll take a look at a history overview of human coronaviruses, the basic structure of human coronaviruses, specifically severe acute respiratory syndrome coronavirus 2, the virus that causes COVID-19, the life cycle, replication of highly pathogenic human coronaviruses, current research for a vaccine for COVID-19, lastly, some global prevention strategies and online resources for COVID-19. So there are 7 human coronaviruses. The 7th most recent is severe acute respiratory syndrome coronavirus 2, SARS coronavirus 2, the virus that causes coronavirus disease 2019, more commonly known as COVID-19. This timeline represents the order and time in which these 7 human coronaviruses were discovered. Similar to SARS coronavirus and MERS coronavirus, SARS coronavirus 2 is an enveloped single-stranded positive sense RNA virus. The genome of SARS coronavirus 2 encodes 4 major structural proteins, including the spike, envelope, membrane, and nucleocapsid proteins. So, highly pathogenic human coronaviruses, such as SARS coronavirus 2, require a host cell to replicate. They enter host cells by first binding to their respective cellular receptor. In this case, that would be angiotensin converting enzyme 2, also known as ACE2. So, SARS coronavirus 2 attaches to ACE2 via the surface spike protein. Then, viral RNA is released and translated into viral polymerase proteins. After this, the negative sense genomic RNA is synthesized and used as a template to form subgenomic or genomic positive sense RNA. Next, the viral RNA and nucleocapsid protein are replicated, transcribed, or synthesized in the cytoplasm, whereas other viral structural proteins, including spike, membrane, and envelope proteins, are transcribed and translated in the endoplasmic reticulum and transported to the Golgi. And then the viral RNA nucleocapsid complex and spike, membrane, and envelope proteins are further assembled in the endoplasmic reticulum Golgi intermediate compartment, also known as the ERGIC, to form mature virion, then released from the host cell to infect more cells. So the spike protein of SARS coronavirus 2 is the primary vaccine target for many candidate vaccines because it mediates host cell attachment and is required for viral entry. So the Moderna vaccine, mRNA-1273, was the first to progress to phase 1 clinical trials in humans. Basically, this vaccine consists of messenger RNA, mRNA, molecules expressing SARS coronavirus 2 contained with lipid nanoparticles to facilitate entry of mRNA into host cells. Once inside the host cell, the spike protein is expressed and induces antibody responses. So, an article published on July 14th of 2020 in the New England Journal of Medicine titled "An mRNA Vaccine Against SARS Coronavirus 2 Preliminary Report" reported, and I'll quote, that the messenger RNA mRNA-1273 vaccine induced anti-SARS coronavirus 2 immune responses in all participants who participated in the clinical trial, and that no trial-limiting safety concerns were identified, and that their findings support further development of the vaccine. Many prevention measures have been implemented by the Centers for Disease Control and Prevention to reduce SARS coronavirus 2 transmission, such as social distancing, mask wearing, hand washing, and contact tracing. Here I have provided some useful COVID-19 online resources. To conclude, 7 human coronaviruses have been identified. Human coronaviruses are obligate intracellular agents meaning they require a host cell to replicate. The spike protein of SARS coronavirus 2 is the primary vaccine target. mRNA 1273 is the first vaccine to progress to phase 1 clinical trials in humans, and global prevention strategies to reduce transmission of SARS coronavirus 2 have been implemented by the Centers for Disease Control and Prevention. In addition to the molecular aspects of COVID-19, I am interested in investigating the COVID-19 prevention measures in underdeveloped countries and how the health and financial impacts of COVID-19 vary across racial and ethnic groups. Thank you to all of the wonderful faculty, staff, offices, and programs that have helped me on this journey. Being part of the CRSP program was a wonderful experience and has opened many doors for me. Thank you again for this opportunity to present at the 2020 CUNY Research Scholars Program Virtual Symposium.